Hey guys, new location unlocked. It's my room. Spoiler alert. I'm just not at school right now, so yeah. Let's get into it. Today we're talking about DNA mutations, so mutations of DNA. La 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 la. First things first is we have a warm-up question. So which of these is the direct result of an error in the transcription of a DNA nucleotide? So to answer this question, there's two things that you need to know. You need to know what transcription is and what is a nucleotide. Okay, go ahead and try and answer the question. All right, so transcription, as we all should know because we've done this so many times at this point, is going from DNA to RNA, and then translation is RNA to protein. Okay, a nucleotide, if you didn't remember, I wish I had my little DNA thing. I love it. Just picture that I have my DNA molecule that I always use. A nucleotide is going to be a sugar, a phosphate, and a nitrogenous base because a nucleotide is the building block of the DNA molecule. It is the monomer of the DNA molecule. Okay, so which of these answer choices would actually match up with that? Well, let's see. The nuclear membrane is ruptured. Would making a copy of DNA somehow rupture the nuclear membrane? No, that would be very bad. As we know, the nucleus is designed to protect and keep the DNA in its own little home. So if the nuclear membrane ruptured every time transcription happened, that would defeat the purpose. Okay. G says amino acids do not bond to tRNA. First of all, tRNA is involved in translation, so no. H, a codon sequence is incorrect. So the codon sequence is going to be the 3 bases of mRNA. I mean, it's possible. I mean, transcription deals with mRNA, so maybe. Let's see if J is going to be right. Transportation of mRNA does not occur. No. Transportation of mRNA just happens. mRNA leaves the nucleus and goes and does what it needs to do. Transportation does not have to do with the actual sequence or with the transcription of the sequence. So the answer is going to be H, codon sequence is incorrect. And that makes sense. An error in transcribing, so going from DNA to mRNA, would lead to an incorrect codon sequence. Okay, so what exactly is a mutation? So you probably have seen these characters, Mystique, not me, Mystique, Professor X, Teenage Mutant Ninja Turtles, or maybe even, what's that dude? What is he? Spider-Man, even him as well, right? Like that's what we kind of think of whenever we think of mutants. Maybe you might have think even of, I think, Umbrella Academy. I haven't seen it yet, but I've heard lots of good things about it. So what exactly is a mutation? We tend to know what a mutant is, but what exactly does it mean in terms of the science behind it? Well, to talk about that, we're gonna have to go back into our discussion of central dogma. And again, central dogma is the law of life. So how your DNA actually codes for and gives you your traits. So DNA, double-stranded DNA, which has T's in it, is going to be copied into mRNA, which is a single-stranded molecule. Instead of having thymine, it's gonna have U's for uracil. That mRNA is then read in groups of 3 called codons, and each group of 3 bases is going to code for a single amino acid. After, or as the cell, as the ribosome is reading the mRNA, it is translating it into amino acids, and those amino acids get all together, they fold all up, and then boom, we have a protein that goes out into the cell and does whatever it needs to do. So this process, however, is not perfect. It's not perfect. You would think it's perfect, but it's not. Sometimes there's mistakes that are made. There's mistakes in transcription, there's mistakes in translation, and there's even mistakes in DNA replication, which is just when DNA copies itself over and over and over again. So, and it makes you, sometimes you might think like, well, how can it really make a mistake? Well, think about yourself. If you were typing a message, or even when you guys answer like attendance questions or your short answers, you might accidentally strike the wrong key. Just as you might make a mistake while typing, while texting, cells sometimes make these mistakes during replication, transcription, or translation. They just sometimes make a mistake. Same way that you do, your cells can do that as well. They sometimes can make a mistake. Sorry, I got a text. So notice right here, this is a changed letter. This right here is an extra letter, and this right here is a deleted letter. That's a hint as to what's to come in the lesson. So again, cells can sometimes make mistakes too. These mistakes are pretty rare, and cells actually have built-in repair mechanisms. Think like, when you're texting and you make a mistake, autocorrect might catch it. Sometimes autocorrect has no idea what you were trying to say, right? So sometimes there's a built-in repair mechanism that can detect and correct most errors. And sometimes there's a permanent error that occurs in the DNA, RNA, or whatever. That permanent error, that permanent change in the base sequence is known as a mutation. So mutation, its definition is very, very simple. A mutation is known as any permanent change in the base sequence of DNA. So here's a good graphic that I really like. So let's say that we have a strand of DNA. We're looking at a specific gene and at its sequence. Okay, this is its sequence. This is going to code for a normal functioning amino acid, which codes for a normal functioning protein, which is going to give us some sort of normal characteristic. However, if a mutation were to occur, such as right here, as you notice, the sequence is different. There's lots of different bases in here. This is the wrong sequence. The wrong sequence is going to code for the wrong amino acids, which is going to make the protein fold up wrong. It's going to be the wrong shape. And if it's the wrong shape, then it's not going to be able to do its job correctly. So it's going to have a loss of function, or maybe a changed function, or something like that. Okay, so that's how mutations actually occur. So for example, everyone's DNA codes for their skin tone, right? Everyone's DNA codes for a normal pigment in their skin tone, a normal amount of melanin depending on what your race and background are. Okay, so but Mystique has blue skin. How could DNA mutate to give someone blue skin? Well, this is just something that I just thought of. So for example, Mystique's DNA might have mutated so that instead of coding for the normal melanin skin tone protein that we all have in our bodies, even white people have it, they just have less of it, then her DNA could code for blue skin tone protein. So instead of coding for melanin, which is just varying degrees of like brown and tan, her DNA might code for blue pigments. That's just an example of it. Ninja Turtles could have some sort of mutant DNA that gives them giant muscles and makes them grow big. I don't know. Just, yeah. Mutation is any permanent change in the DNA sequence.
The differences in our genes come about through a natural process called mutation. Mutation is extremely common. As a cell copies its DNA before dividing, it makes occasional typos. Most commonly, a single base is substituted for another. Sometimes a base is deleted, or an extra base is added. The cell is able to repair most of these typos, but each round of cell division introduces a few changes that are not repaired. When DNA changes occur in cells that will give rise to eggs or sperm, they can be passed down to offspring. Each one of us has about 60 new variations that were not present in our parents. People commonly use the terms mutant and mutation to describe something undesirable or broken. But mutation is not always bad. Most DNA changes fall in the large areas of the genome that sit between genes, and they usually have no effect. When variations occur in areas of the genome that code for proteins, they often have consequences. These changes can affect the protein product itself, or they can affect when, where, or how much protein is made. Most of the time, mutation generates variations that are neither good nor bad. Just different.
We're going to keep it going. So, can mutations ever be helpful? Because we usually think of mutations as bad or harmful, but can they ever be good? And I'm not talking about like Spider-Man saving the day. I just mean like in real life. Can mutations ever be good? Well, possibly. Mutations are just a source of genetic variation. If you remember from our discussion of genetic variation, that just means the diversity of life, the diversity of different organisms within a population or within the entire world. So again, a mutation is going to be a random change in the base sequence. Any random change. The random change can have a bad effect, it could have a good effect, or it could even have no effect on the organism. Okay, mutation doesn't inherently mean bad, good, or anything. It just means that there's some sort of change somewhere. That is what a mutation is. Any change at all. Doesn't mean it's bad, doesn't mean it's good. Okay? In populations, mutations actually can be kind of good because they can give rise to diverse and cool organisms. So for example, did you know this? Blue eyes are a mutation. Humans did not originally have blue eyes. Okay, you can look it up if you don't believe me, but yeah, blue eyes are a mutation, actually. Doesn't mean that everyone who has blue eyes is a mutant, but the original people did not have blue eyes. That actually is a relatively recent mutation in terms of the longevity of human history, or not just of human history, of the history of the earth. Okay, mutations can also help out. So let's say this is a normal, the normal, which is also sometimes said as wild type. The wild type characteristic doesn't allow this organism to reach this food. However, an organism that was born with a mutation in their DNA might have had a mutation that makes the proteins in their beak grow longer and pointier. If it grows longer and pointier, then it's actually able to reach more food. That actually can be a beneficial mutation. Okay? There's also mutations that can help us grow food and grow medicines and things like that, such as seen here with corn. This actually is a mutant corn that they're saying could solve world hunger because it can grow in such easy conditions. So again, mutations can be beneficial, harmful, or even neutral. So I don't know, I feel like a lot of people don't know this, but humans and other mammals, we're supposed to really only drink milk when we're young, like when we're like babies. But we basically have forced ourselves to keep making this enzyme called lactase, which again, enzymes end in ASE. And essentially all it does is it breaks down lactose, which is found in milk. But since we have forced ourselves to keep drinking milk, essentially we have just forced ourselves to keep drinking milk, even though, I mean, it's not really feasible. That's why in a lot of places that are not like near America or like Americanized or affected by America in a lot of ways, they cannot drink milk. They do not have the lactase enzyme because they don't have it in their genome. Okay, I was gonna move on from that. So again, this is the process of central dogma. So we have DNA, double-stranded DNA. It's transcribed into a single-stranded copy. That single-stranded copy, that messenger RNA, gets to leave the nucleus, finds the ribosome, and then it gets translated into a protein. Okay, just so here's the same thing. Nucleotide sequence determines the amino acid sequence, which determines the protein shape, and the protein shape determines the protein's function. I don't know what this is. Oh, yes, another graphic. So we have a normal gene. That normal gene codes for normal mRNA, which codes for normal amino acids, which makes a normal protein, which has a normal function. While a mutated gene creates mutated mRNA, which creates mutated amino acids that either, in really bad effects, can have no protein, or there also can be abnormal proteins that do the wrong job. Okay, this is seen in certain types of diabetes where people do not make insulin protein, which means they cannot process their body sugar. That is due to a mutation in their genome, most of the time. So, fact, many people with breast cancer have BRCA1, and so you say that BRCA1 proteins that are missing, or that are wrong shape or are missing amino acids. How does that happen? Like, how does that happen? Well, there's a mutated DNA sequence somehow. Then the incorrect amino acids are made. If the incorrect amino acids are made, then the protein structure is wrong, which means the protein's function is wrong, and that's what gives the symptoms of the disease that are seen. Okay, that's just an example of how that affects you in real life, how it can be related to real life. Cool. So types of mutations. We're going to talk about point versus frame shift, and then we'll talk about gene or chromosomal mutations, because there's different types of mutations. As is everything in biology, all these different types. Okay, so a point mutation is going to be when one nucleotide is switched out for another. And if it isn't fixed by the cell, it usually only results in the change of one amino acid. So at one point, the mutation occurs. That is a point mutation. right? At one point along the DNA or the mRNA or whatever it is, it's mutated. One nucleotide was accidentally switched out for another. Sometimes this can be called a substitution mutation. Why do you think that? Well, a substitute just means to switch. So for example, if there was a substitute in one of your classes, your teacher just switched with another teacher or with a substitution mutation. So point mutation, substitution mutation, same thing. And if you think about what the words mean, it makes sense. Okay, then we have what's called a frame shift mutation. That's when one nucleotide is inserted or a nucleotide is deleted. The entire amino acid sequence is changed, resulting in incorrect proteins being made. Why do you think this is called a frame shift mutation? Well, we know that we read DNA and RNA in bases of 3, basically, right? The codons in the RNA. So if I insert one here, then suddenly everything has to be shifted. Don't believe me? Okay. Don't worry, we're going to prove it. So think about this. What type of mutation, point or frame shift, would you say has a more negative impact on the protein product? Let me move myself so you guys can look at the picture. All right. So a point mutation is going to affect only a single amino acid. That's going to barely affect the protein structure, and that barely affects the protein's function. However, a frame shift mutation is going to affect every amino acid after the mutation. That's going to heavily affect the protein structure and function. And again, if you don't see how, you don't believe me, let's look at some examples. Let's prove it to each other. Let's prove this concept to each other. So here's a sentence I made up. I don't think I made it up. I think it's just a sentence that we use to teach this concept to students. The fat cat ate the rat. A sentence that makes sense. Note how the letters are sequenced into groups of 3, like how we read codons. This is known as a 3-letter reading frame. This reading frame determines the exact sequence of amino acids that will be made for this protein. Amino acid 1, amino acid 2, 3, 4, 5, and 6. This is what's known as the reading frame because you read it in groups of 3. That codes for one amino acid, that codes for another one, and so forth. Now let's add the letter T between the words fat and cat. So we're going to do an insertion. We're going to do an insertion right here and see how that affects the amino acids. Okay, so this is one of the major mistakes that students make. They just insert that base and then they're like, okay, well, this is all fine. No, we read them in groups of 3. Our reading frame is always in groups of 3. So we start from the beginning and we say, okay, 1, 2, 3, boom. 1, 2, 3, boom. 1, 2, 3, boom. That is one amino acid. Then the next 3. That is another amino acid. Then the next 3 amino acid. And then the next 3. Okay, we always read them in groups of 3. So now I've adjusted it to where it's in groups of 3. Does this sentence make sense to you? Would this protein product be functional? No. Inserting a base caused the entire reading frame to shift. As you can see, it's all shifted backwards, right? We see the, fat, and then we see cat, ate, the rat. The reading frame is now shifted. It's just moved over. So instead of seeing the correct words, we now have a non-functioning product. So insertions definitely affect almost the entire protein. Okay, now we're going to start back from the same original sentence, and now we're going to remove a letter. We're going to remove the A in 8. We're going to remove this letter, and then we're going to see what happens to the sentence. Okay, so I've removed it, as you can see, but can I just go ahead and look at it now? No, we have to move them into groups of 3. So 1, 2, 3. That's a codon. 1, 2, 3. That is a codon. 1, 2, 3. That is a codon. 1, 2, 3. That is a codon. 1, 2, 3. These are a codon. So here's what it looks like. What effect did removing the A have on the sentence / protein product? Is it functional or not? The fat cat was understood, but the rest of it is pretty much jumbled up. Okay, the protein is messed up. Now, here's something that I didn't think about when I was making this, but I've just noticed this. This right here is a word, so that does actually kind of make sense. And that is a special type of mutation in which a different amino acid is made, but it could still kind of make sense. Okay, but we'll get into that in a little bit. The fat cat ate the rat. Back to our same original sentence, but this time we're going to change this C into a B. However, before we do that, what type of mutation would that be? Is that a point mutation or a frame shift mutation? That's going to be a point mutation, aka substitution mutation, because we're going to substitute this C for a B. There we go. What happened to our protein product? Is it functional or not? The sentence still makes sense. And this is the proof. A substitution mutation often only changes a single amino acid. That one amino acid might not have an effect on the protein folding. Thus, it's not that big of a deal as compared to frame shift, insertions, and deletions. Right? When we inserted or deleted letters, we had to change everything around. But if we have a substitution where there's one base was accidentally switched out for another, it doesn't affect the protein that much. Okay, we see it? Okay. So this is a normal DNA sequence. Now in the substitution, as we notice, these were switched out. A deletion, that was deleted, and an addition, those were added. This is what it could look like on an actual DNA molecule. Okay. There's many different ways to model this. So let's just review what we've just learned. Substitution mutation is when one nucleotide is replaced with another one. So for example, the cat ate the rat versus the bat ate the rat. Insertion mutation is going to be when one nucleotide is added. For example, the cat ate the rat versus the akadawrado. Notice that didn't make sense. Deletion is when one nucleotide is removed. So the cat ate the rat versus the cat doradaw. Again, that didn't make sense. Okay, here's another graphic of what that could look like. So gene insertion is when Uncle Guanine makes an uninvited and extended visit. See, they're doing all good and then suddenly, boom, he's in there. Bases can sometimes some get inserted because DNA polymerase and RNA polymerase are error-prone things. You'll learn that in not high school, college biology. Okay. I really like this graphic. So the normal thing could be beast. A substitution mutation could change it to feast. An insertion mutation, when you insert a letter, could be breast. Deletion mutation, best. Or an inversion, which is one that we don't really talk about that much. But inversion just means to switch the two. Which obviously means this doesn't follow our codon rules, but this is just like for demonstration purposes to make sure that you know the concept of what those mean. Again, here's another graphic of what mutations can look like. Okay. What type of mutation would this be? Going from here to here. What type of mutation would that be? Okay, good job. So that would be a substitution mutation. Okay, going from here to here. What type of mutation would that be? insertion, because it's inserted. Okay, and then here, what type of mutation would that be? Deletion, because the base was deleted. Good job, good job, good job, good job. Okay, mutations can also be classified on how they actually affect the amino acid. So these terminologies are used somewhat less frequently, but I still want you guys to know them. So a silent mutation is when between the original sequence and the mutated sequence, somehow the exact same amino acid is made. That's known as a silent mutation. How does that happen? Well, there are multiple, if you look at a codon chart, there are multiple ways to make the same amino acid. Okay, a missense mutation is from between the original DNA sequence and the mutated DNA sequence, a totally different amino acid is made. And then a nonsense mutation is when between the original DNA sequence and the mutated DNA sequence, that suddenly a stop codon is made in the mutated one. Okay, I always remember the difference between these two because nonsense has an O in it and so does stop. That's how I have to remember it. Okay, and this is what this can look like. So, if the original DNA codon, or the original DNA says UUA, which this wouldn't be DNA, actually would be RNA. Not my picture. UUA, that's going to correspond to the amino acid leucine. A frame shift mutation would be if there was an insertion, and that's going to affect every single amino acid after it. Okay, if the original was UUA, and suddenly it was changed to CUA, well, if you look on a codon chart, that still codes for leucine. Okay, a missense mutation would be if it was UUA, which is leucine first, and then suddenly it was changed to GUA, which if you look on a codon chart, that's going to code for valine, which is a different amino acid. So that's a missense mutation because a different amino acid was made. And then nonsense mutation is going to be, let's say if it was UUA and then suddenly it was changed to UAA. If you look on a codon chart, that codes for the stop codon, for a stop codon. So will the mutation lead to biodiversity? Well, first things first, we have to talk about the difference between somatic cells and gametes. So a somatic cell is going to be any of your body cells. So your skin cell, liver, eyeballs, muscles, heart cells, all of that. Basically 99% of the cells in your body. Versus gametes are a special classification of cells that are just known as the sex cells. Eggs and sperm. That's it. If it's not an egg, if it's not a sperm cell, then it is a somatic cell. Period. Okay? Clearly, we all should know this, that sperm and egg fuse together, they fertilize, and that is how a baby is made. Versus somatic cells are just your body cells. So if a mutation occurred in a random somatic body cell, would a mutation here get passed down to offspring? How about if it occurs in sex cells, such as sperm or egg? Would the mutation get passed down to the offspring? Well, if it occurs in a somatic or body cell, no. Because random body cells, your eyeball cells, your hair cells, those are not involved in procreation at all. They're not involved in procreation. So if a mutation occurs in them, it's going to occur in that cell and that cell only. It's not going to affect your children in the future. Now, if a mutation occurs in sex cells, such as a sperm or egg, and then someone procreates with that mutated sperm or egg cell, then yes, of course, the new baby is going to have whatever mutation was present in the sperm or egg. Okay, I feel very passionate about this because this is a question that always comes up, and I get so sad whenever students get it wrong, because the only way that it can be passed down to a new baby is if it occurs in a sex cell that creates the new baby. Okay. Also, this is tying back into something we learned at the very beginning of the year. Mutation is one source of genetic variation. that actually can help you that actually can help make you different from your parents and different from your siblings, even though you guys share a lot of the same DNA. Okay, random mutations can sometimes help. Okay, so we're pretty much done with the lesson material. So now we're going to do a couple practice problems to see what we know. So a certain mutation in the gene for hemoglobin results in red blood cells becoming sticky, rigid, and irregularly shaped. These irregularly shaped red blood cells block the flow of blood throughout the body. A single base mutation is responsible for these irregularly shaped blood cells. So here's what it looks like. So again, we have this normal gene, which has normal DNA, which makes the normal mRNA, which makes the normal protein, glutamine. Then we have a mutated gene that's going to cause a mutated mRNA, as you can see. The normal sequence is GAG. The mutated is GUG. If you look on your codon chart, that's going to code for valine, which is a different protein than it's supposed to be. Okay, so which of these mutations most likely results in the mutated hemoglobin gene? Would you say this was an insertion, a deletion, a duplication, or a substitution? Well, look right there. GAG to GUG. Were any bases added? No. The A was actually substituted for the U. They just were switched. Okay. Boom. Okay, next question. No, not two. That's the problem number. Different types, I'm sorry that I'm a mess. I'm sorry, okay, I apologize. Different types of mutations can occur in DNA. This diagram is going to represent a certain type of mutation. So we have a chromosome, we're going to have part of this DNA that we have right here. Here's a sequence. Then there's some sort of mutation, and then here's a sequence with the mutation. Which statement describes the mutation in the diagram? So here's the sequence written out. So GAC, TAG, CAC, GAC, TAA, GCAC. So I mean, when you write it out like that, that's why you always write it in groups of 3, because you can easily see it. So a base was definitely inserted into one strand of the DNA. As you can see, this right here was an insertion, because it's G, and they both have A, and they both have C, they both have T, they both have A. Oh, there's a random letter here, but let's just keep going to see. G, C, A, C. So the only thing that is different is this one right here, which is an insertion. All right, so I hope that despite my shortcomings, we understand mutations, we understand how they work, and how they happen. Shout out Spider-Man and Professor X. Okay, goodbye.